Do you know how fireflies, foxfire fungi, and messenger RNA vaccine are related? That's right, if you guessed it, it's bioluminescence. Hi, my name is Robert, and together with James and Allison, we will be presenting on bioluminescence and luciferin. So before we delve into the details of bioluminescence and luciferin, let's describe a brief history of the phenomena. Aristotle was the first one to record bioluminescence, and he described a cold light which was observed in marine species. Christopher Columbus had a similar encounter, describing a burning sea on his journeys. And William Shakespeare describes an ineffectual fire of a glowworm in his work Hamlet. And then in Cuba, during the colonialization period, English explorers avoided landing on the island due to mistaking light from fire beetles to be Spanish campfires. Ultimately, Raphael Dubois coined the term for the words luciferin and luciferase, which derive their origins from Latin meaning light bearer. So the difference between bioluminescence, fluorescence, and phosphorescence, I think, is necessary to establish just to reduce any confusion. Bioluminescence occurs when a cell within a cell and produces light as a result of chemical reactions. Fluorescence occurs when a substance absorbs light from a source that is then re-emitted. And phosphorescence is like fluorescence, however, the re-emission period is long-lasting following the exposure to the initial light source. The image below, I think, describes it very well. So in the case of fluorescence and phosphorescence, the excitation is due to a photon, and then in bioluminescence, the excitation is due to a chemical reaction happening within the cell. Bioluminescence occurs as a chemical interaction between luciferin and luciferase to produce photons of light. Bioluminescence has evolved independently about 30 different times, and that's across a variety of species across the domains and kingdoms of life, including bacteria, fungi, plants, and animals. Luciferase can alter color depending on its expression, and secondary emitters and proteins can also cause a color shift. One study suggests that a single amino acid change in luciferase can alter the color expression in luciferin. Ultimately, bioluminescence is responsible for camouflage, defense, predation, and communication within nature. However, in the case of bacteria, it's still unknown how and why bioluminescence evolved. One study suggests that it's just an indicator that there's oxygen present, but it's still open to debate on the details. Given the fact that bioluminescence has occurred independently 30 different times, luciferin is more of an umbrella term for multiple molecules that produce light, that are light bearers. And of the ones displayed on the screen right now, they occur in freshwater snails, bacteria, fish, shrimp, single-celled eukaryotes, deep-sea fish, and fungi. So there's a whole diversity that occurs just on these molecules on this slide right here. Coelenteriazine is used in some fish, shrimp, and other marine species, and it produces a blue light shift due to a protein called aquarin. So the reaction between luciferase and luciferin is not one-dimensional. It can produce a whole spectra of colors. And then hispidin is seen in the foxfire fungi that I mentioned at the very beginning of this presentation. Of the molecules just presented, the most important one would be D-luciferin. It's found in fireflies and it's used in a lot of research, and so it's the most well used luciferin molecule. And it's used as a reporter in assays and in bioluminescent imaging. So in assays, it's used as a reporter molecule. So when you're trying to translate a specific protein, oftentimes the messenger RNA is encoded with the target protein and luciferase, and then to confirm the presence of the translation, luciferin will be introduced, and if there is any luminescence seen, then that indicates that the luciferase was translated, meaning that the target protein is also translated. And then with the design of the COVID messenger RNA vaccine, it took a similar approach. In order to get the messenger RNA vaccine into the cell, it needed to be transported through the body and not be degraded. So to do that, it needed a transport vessel, which came in the form of a lipid nanoparticle, which operated as the delivery system to the cell to protect the messenger RNA from degradation, within the body. So in this particular study, based off the image on the right, different lipid nanoparticles were used to test out which is the best delivery system. And to report the results of this delivery system, luciferase was used in mice. So the messenger RNA encoding luciferase was placed in one of these lipid nanoparticles, and then it was introduced into the mice in 3 different routes. And then shortly after that, luciferin was introduced into the mice as well. And the luminescence was then recorded, and the higher the luminescence indicated the higher the expression of the luciferase enzyme, meaning that the lipid nanoparticle was operating with the greatest amount of efficiency whenever there was the highest degree of luminescence. And so as you can see on the image on the right, the lipid nanoparticles number 14 and 15 were consistently producing a great amount of luminescence, bioluminescence, throughout the 3 routes of intramuscular, subcutaneous, and intradermal. And so this information helped make the design for the carrier for the messenger RNA COVID vaccine. Now, D-luciferin does have its drawbacks in terms of bioluminescent imaging. D-luciferin can poorly cross the blood-brain barrier, so it does not accurately represent the expression of luciferase within neural cells. So to get around this, there is a synthetic luciferin that is currently starting to become introduced, in particular with these bioluminescent imaging studies. And it can cross the blood-brain barrier better than D-luciferin, so it can more accurately present the amount of luciferase present in brain cells. So there are some limitations to luciferin. However, it is currently one of the most well understood and well used molecules of luciferin that is used in research currently. And James and Allison will go into greater detail about D-luciferin and how it operates and how it's regulated as well.
So, as Robert was saying, this D-luciferin molecule is what fireflies use to emit their bioluminescence. And this is a very basic overview of how this process works, but essentially, the luciferin will bind to the luciferase enzyme, and in the presence of oxygen and ATP, it will undergo an oxidative step to be excited to this singlet excited oxyluciferin molecule. And when it returns to its ground state, it emits this energy in the form of light, which is the bioluminescence that we can see and observe. Now let's see how this process really works. The first step in the luciferase enzyme mechanism requires the presence of ATP. And one of the foundational features in biochemistry that makes ATP such a good molecule to attack is this presence of this magnesium ion, which stabilizes the beta and gamma phosphates, allowing for the alpha phosphate to become even more electrophilic, making for easier nucleophilic attacks, which is exactly what happens here. The carboxylic acid on luciferin is going to undergo an SN2 nucleophilic attack on the alpha phosphate of ATP, kicking off this pyrophosphate molecule, which is then further stabilized after it's kicked off by magnesium. And what we're left with is an AMP molecule bound to luciferin. In the second step of this mechanism, we see the importance of the AMP molecule being attached because it creates this acidic region, more specifically this acidic hydrogen on carbon 4 of the ring on luciferin, that allows for a base in the active site to come in and deprotonate this hydrogen. And what this does is create a carbanion species, which in the presence of molecular oxygen can attack the molecular oxygen, resulting in the two oxygens being attached to that carbon and ultimately resulting in this hydroperoxide species characterized by the attachment of molecular oxygen. And this is considered the oxidative step of the reaction. The third step in this mechanism is characterized by a nucleophilic acyl substitution by the hydroperoxide. Now, it attacks this carbonyl on the luciferin molecule because carbonyls are susceptible to attacks due to being somewhat electron deficient from the pull of electrons heavily towards the oxygen. And what this does is pushes electrons up onto the oxygen, creating a negative charge and creating a tetrahedral intermediate, which we see right here. And to complete the nucleophilic acyl substitution, this tetrahedral creates a favorable interaction to kick off the AMP leaving group, which is a good leaving group in this case. And it does this by pushing the electrons back down into the tetrahedral and then eventually kicking off the AMP molecule, leaving us with an alpha peroxylactone, which is high energy, and it's called a dioxetanone. The third step in this reaction is characterized by the nucleophilic acyl substitution of the hydroperoxide. And we see that here when the hydroperoxide attacks the carbonyl group of the luciferin molecule, which is susceptible to attack because it's somewhat electron deficient from the pull of electrons heavily towards the oxygen. And so when this hydroperoxide attacks, it pushes the electrons up to the oxygen, leaving it with a negative charge and forming a tetrahedral intermediate that we see right here. So to complete the nucleophilic acyl substitution, this tetrahedral sets up a perfect opportunity to kick off the AMP leaving group by just pushing the electrons back down into the luciferin molecule, into the tetrahedral, and kicking off this favorable AMP leaving group, which leaves us with a alpha peroxylactone, which is actually a high energy intermediate called a dioxetanone, and it's a conserved motif across several chemiluminescent systems. Now, this next step of the mechanism in luciferase involving the decomposition of dioxetanone is subject to some debate, but can be explained through radical chemistry, and that's what we see here. Beginning with the electrons of this oxygen through resonance can be pushed all the way to this nitrogen, adding a negative charge to that nitrogen. This favors a radical chemical reaction between this high energy peroxide bond in which homolytic cleavage takes place, where one electron is distributed to this oxygen, one electron to this oxygen, and the nitrogen actually donates an electron to the oxygen on carbon 4, giving it a stable 2 electrons, but leaving the nitrogen with radical chemistry, ultimately resulting in a diradical species, one singular electron being on this oxygen and one electron on nitrogen. Now, this diradical species that's formed is extremely unstable and will undergo a spontaneous decarboxylation, in which an electron from this carbon to carbon bond is donated into the unstable radical oxygen, where that radical electron can be donated in to form a much more stable bond, while also donating the other electron into the luciferin molecule, where the carbon 4 of the luciferin molecule of this ring, where the this portion can be kicked off as CO2, and which opens up a favorable reaction, which involves the electrons from this oxygen being donated to form a much more stable double bond and stabilize this nitrogen by combining these radicals into a much more stable negative charge. And this is what ultimately brings us to this singlet excited oxyluciferin, represented in green, high in energy and anxious to release it. Now, in the final step of this luciferase enzyme mechanism, it's important to note that what's being depicted is this singlet excited oxyluciferin in the keto form. It can also be present in the enol and enolate form. Now, just like we saw before with the electrons from this oxygen, that's where they'd be located. The electrons from this oxygen being passed all the way through to the nitrogen through resonance. Well, the same exact thing is going to happen as this molecule enters its ground state, or is directing towards its ground state, when the nitrogen is going through resonance, past the electrons all the way back into the oxygen, creating what we saw before, where with the electrons from the oxygen over here and a stable nitrogen molecule. Now, in this excited state of this this oxyluciferin, this anionic oxyluciferin, when it returns to its ground state, it releases the energy in the form of bioluminescence, and what has resulted is the ground state luciferin molecule that we begin with. And this is the overall process of how fireflies utilize luciferase to emit bioluminescence. So Allison's going to talk a little bit more about biosynthesis and regulatory pathways of the luciferin molecule.
I am presenting the biosynthesis and regulation of luciferin. Again, this is the structure of the luciferin molecule present in the firefly and the beetle, which is the most thoroughly studied luciferin. In regards to its biosynthesis, there is no evidence of the direct ingestion of the molecule by the firefly or the beetle, so it's predicted that the molecule is synthesized in vivo. Based on the structure, it is likely that the thiazole ring on the right originates from a cysteine amino acid when we look at their similar structures. This is the predicted synthesis of luciferin from benzoquinone. It was an early attempt of the synthesis of luciferin in vitro, which can be used as a model for the synthesis of luciferin in vivo in the living organisms, but it is not proof of how it is actually synthesized in the firefly and in the beetle. This synthesis was predicted based on the possibility of quinones being present in the beetles, as they're used in the their defense secretions. In this example, benzoquinone reacts with cysteine to create luciferin. This is the predicted synthesis of a different luciferin molecule present in the sea firefly, which is a crustacean. The structure was obtained from mass spectrometry, and I included it because it shows a technique to predicting biosynthesis. There's a technique where when you look at the structure, if you see certain amino acid moieties, that's a good starting point to synthesis of the molecule. And in this one, you can clearly see aspects of isoleucine, arginine, and tryptophan to create the luciferin molecule. This is the predicted synthesis of luciferin from oxyluciferin. As we recall, oxyluciferin is the product of the reaction of luciferin with the luciferase enzyme. So this synthesis of luciferin is a recycling of that product back into luciferin to generate more light. In a 1974 study in vivo, which compared to the last predicted synthesis, which was in vitro, scientists injected tagged oxyluciferin and 2C6HB, which is this intermediate 2-cyano-6-hydroxybenzothiazole, into live fireflies, and they detected tagged luciferin over time, which suggests that the oxyluciferin and 2C6HB were recycled back into luciferin. The addition of cysteine sped up this reaction, and it was indicated that it was a two-step reaction. So in this first step, it is the transformation of oxyluciferin to this 2C6HB intermediate, and then the condensation of this intermediate with cysteine to yield luciferin. As a precursor to an explanation on a later slide, the chirality of the cysteine added determines the chirality of the luciferin molecule itself. This is the same predicted regeneration pathway, just shown differently by a different source. This utilizes the luciferin regenerating enzyme, LRE, which is circled in this right arrow, which is not well studied. But it is predicted that LRE converts oxyluciferin to the 2C6HB intermediate. This is the overall reaction of the luciferase enzyme, which is luciferin reacting with one molecule of ATP and one oxygen to generate an AMP, pyrophosphate, carbon dioxide, oxyluciferin, and a photon of light energy. Studies have shown that the light intensity generated is proportional to the concentration of ATP. This is used in cancer studies as light intensity tests signal the age of cells, because as cells and mitochondria age, they generate less ATP, indicating lower light intensity. And in regards to the regulation of this luciferase enzyme, high levels of AMP, pyrophosphate, carbon dioxide, and oxyluciferin inhibits the reaction as they are products. And as an important note, pyrophosphate is a high energy product, which makes the reaction favorable in the forward direction. As another important note, a magnesium +2 ion is a required cofactor with the ATP. This is just the first step of the luciferase reaction, highlighting magnesium as a cofactor. The magnesium, as shown in here, partially shields the negative charges on the oxygens of the phosphate, stabilizing the ATP and promoting nucleophilic attack, assisting the leaving pyrophosphate group. Back to the luciferase reaction, we can see that the chirality on the luciferin molecule, as shown with the arrow on the chiral carbon, depends on the cysteine used. But luciferin is found as either a D or an L isomer. However, only the D-luciferin, as shown in this reaction, reacts in the biochemical pathway to produce light. L-luciferin acts as a competitive inhibitor, and both can be adenylated by luciferin in the first step. But how do we account for that chirality of the luciferin molecules? As we recall, the luciferin synthesized from cysteine, that it's the chirality on the cysteine used that determines the chirality on the luciferin molecule. But in nature, only L amino acids occur in proteins. So where would the D-cysteine come from? And this reaction shown in this slide, we begin with the intermediate, the 2-cyano-6-hydroxybenzothiazole, reacting with L-cysteine, the cysteine that occurs in nature, to form L-luciferin. There is an intermediary adenylation and then reaction with coenzyme A to generate luciferin-CoA. The keto-enol equilibrium of L-luciferin-CoA, as shown in this bracketed fourth step, leads to the L and the D isomers of the L-luciferin-CoA and the D-luciferin-CoA. These two are then hydrolyzed to form the L-luciferin and the D-luciferin. So overall, it's just synthesized that the intermediate, the 2C6HB interacts with L-cysteine, is adenylated, interacts with CoA, and then it is keto-enol transformed over into a D-luciferin-CoA, and then hydrolyzed to D-luciferin to react with luciferase. This is the same luciferase reaction equation, but with other side reactions included. As shown in this graph on the right, with in vitro bioluminescent light emissions, there is an initial flash of light that decays to a standard basal level. So which indicates that yes, the luciferin does bind to luciferase, but there is a competitive inhibitor that also binds to luciferase. Studies have shown that LAMP, which is the dehydroluciferyl AMP, is the strongest inhibitor of luciferin and acts as a tight binding competitive inhibitor. There is also competitive inhibition of the luciferase molecule by long chain fatty acids, as they are substrates of luciferin.